Hello AP Biology students, we're going to go over the second section in Unit 3. And in this section, we're going to go over something called enzymes. Enzymes are proteins that facilitate chemical reactions. And a really important aspect about enzymes is they are reusable. If you remember from our last section, enzymes are called catalysts because they help speed up chemical reactions and aren't actually used up in the reaction. In science, we tend to name enzymes by what they react with, and we add the ase suffix to the end of the enzyme name. For example, like amylase breaks down amylose, or lactase breaks down lactose. This isn't always the case. You can see here like trypsin and pepsin, they break down proteins, but they relate to what it actually is going to react with. Now, I like to tell students that there are two main functions that an enzyme can complete. Enzymes either join molecules together or break molecules down. Again, this is a generalization. There are enzymes that can do maybe other things or maybe even do both, but typically enzymes do one of those two things. They join things together or they break things apart. There are a couple of vocab words that we have to know when going over enzymes, and the first is a substrate. Substrates are the reactants of an enzymatic reaction. Basically, substrates are what goes into the reaction. The active site is going to be the location on the enzyme where the substrate is going to bind to. As you can see here, our enzymes in blue, and the active site here is where the substrates are going to bind to. Whenever the substrates and enzymes come together, it's going to form something called the enzyme-substrate complex. This is where the enzyme is going to facilitate some reaction with the substrate. Whatever is left over or is produced is going to be called the product. So again, we have our enzyme, our substrates, our active site where the substrate meets our enzyme. When they come together, it's going to form the enzyme-substrate complex, and the substrates are then going to be changed into the product. Fantastic meme. Enzyme looking at its substrate like, I'm going to get you. An important aspect about enzymes is they have a 3D shape. And a lot of times what we show when we're talking about enzymes is a two-dimensional figure like you see here. This creates a misconception where students think that enzymes are flat, and they're not. Again, they have a three-dimensional shape. They're also going to be specific. Typically, enzymes are going to only react with one type of substrate. The substrate an enzyme reacts with is going to be based on that enzyme structure. Remember that structure determines function. That's a major theme within AP Biology and just science in general. So the enzyme's structure or shape is going to determine what type of substrate it reacts with. Classic Drake meme here. Now, one thing that we add into our understanding of enzymes when it comes to AP Biology is the understanding of something called induced fit. This is that next layer deeper of what actually goes on within enzymes and their enzymatic reactions. Basically, what happens is the enzyme isn't going to be perfectly matched up with the substrate, which means it's not going to be a perfect match. You can see here we have the enzyme and the active site, and it actually doesn't look like it's going to bind with the substrate. What has to happen is a conformational change to allow that substrate then to fit. This conformational change will in turn then change the substrate in some way, either breaking it apart or joining them together. So basically, that shift where we see the enzyme just kind of making a conformational change or changing shape is going to be what is actually causing the reaction with the substrate. This is again called the induced fit model. Perfect meme here. Perfect.